So our development started from the understanding of a PSA and a PSMA in association with prostate cancer. And we took this, our technology advantage using the SYNCON technology we call SYNCON technology, which is unique optimized DNA plasma expression vector to incorporate of a PSA and a PSMA and into that's we call DNA vaccine. And you can see there is similarity across different species. And we're using this as an immunotherapy targeting for the clinical indication, specifically for prostate cancer in this setting. As we also know, the DNA transfection have big challenging about how to increase the efficiency of a cellular transfection. So we have our unique technology to deliver the DNA through the electroporation in vivo and to increase the cell membrane permeability to allow the DNA molecule enter to the target cells. And you can see we have different, let's see which ones. But you can see from the slides, we have two different type of device. One is intramuscular injection. Another one is intradermal, so we can use that for clinical study. And we tested this our DNA vaccine in mice, and you can see without intramuscular, without IP. So you see this is very small amount of cellular response after injection, but with the electroporation in vivo, so there's a 100-fold increase of DNA transfection as a readout of interferon gamma elaspods. We use the combination of both PSA and PSA and the PSMA DNA vaccine in the normal mice, and generate after two injections and 2 weeks later, and we have the peripheral blood from the treated animal or naive animal, and to read out as cellular immune response. In here, we demonstrate a robust T cell response read by the interferon gamma elaspods, and also by the flow cytometry. In this case, is a T cell specific predominant, and you can see the both the CD4 and CD8 T cells response. We also observe the SYNCON PSA, the PSMA DNA vaccine generate antibody specific for these antigens. And here we are testing in two different studies. One is using the human prostate cancer cell line, and another one is using 3 or 4 Gleason 3+4 patient tissue specimen for this study. And actually, in this particular study, we're using either the normal serum or the animal serum who receive the immunotherapy to staining with either cell line and tumor tissue, demonstrate that's antigen-specific antibody production. Next move, we're using the TRMP-C2 mouse model for tumors to read out with, in this particular slides, I demonstrated the PSMA treatment effect. And after the 4 treatment, and we observed the significant slow tumor growth as well as animal survival in this specific tumor model. We observed a similar observation of induction of robust T cell response in non-human primates. And we combine the PSA and the PSMA vaccine and treated with non-human primates, and demonstrated a significant increase of antigen-specific T cell response, as well as interferon gamma elaspods indicate of the T cell activity. More importantly, on this slide, on the right panel, we also observed the correlation of specific CD8 T cell production of Gramzan B, CD107A, and TBAT, which we consider the indicator of effector cells and CTL. Not only in our preclinical and translational research, we also proved our technology and the platform in clinical trial. In this particular case, I want to bring your attention is this is not in the prostate cancer, but this is just recently we finished the phase 2 randomized double-blind placebo-controlled clinical trial in high-grade cervical dysplasia, which is the SIN2 and the SIN3, and recently we published in the Lancet. In this particular study, we have 167 women have SIN2 or SIN3 condition, which is also associated with HPV 16 or 18 co-infection, and the primary endpoint is disease regression. And our primary endpoint, as I mentioned, is disease regression. So in this study, we demonstrate a statistically significant achievement in the disease progression in the treated patient. We also observe the virology clearance in treated patient as demonstrated here, which is reached statistically significant. In the correlative research part, we demonstrate the correlation of the antigen-specific T-cell response in this particular case. And you see there's a quite significant increase from the baseline. So I want to point out the 0 is the baseline. It now means the patient, the peripheral blood antibody is 0. It's just we using the baseline as 0 as a comparator. Start from that point, you can easily to understand the patient that received treatment or in the naive treatment group, in the placebo control group. You see there is a 100-fold increase in the T-cell response in particular study of interferon gamma elaspots. Similarly, we use flow cytometry. We demonstrate this is a CD8 T-cell predominant effect, and also demonstrate the lytic potential, which we consider is correlated with our clinical outcome. In the tissue sample we collected from the treated patient compared with a placebo-controlled patient, we observe the clearance of viral infection. You can see on the left side panel before treatment and after treatment, which indicate the P16 protein, indicator of the HPV viral infection. On the right side, we demonstrate the CD8 positive cell infiltration into the tumor area, which highly suggests that they may be contributed to the clinical outcome. Right now, we have an open label study using a modified 3+3 design, in the combination with the PSA, PSMA, and the interleukin-2 DNA as adjuvant in the biochemical relapse the patient population of prostate cancer. And this study is multi-center in the US and start enrolling on our schedule. The primary endpoint is safety tolerability, of course, and the immunogenicity. We also try to determine the preliminary PSA response, although this is a PSA rising biochemical relapse patient population, as we understand. We believe this study will give us some benefit of understanding the safety profile of this immunotherapy combination. Also, we will try to determine the optimal dose, as well as to determine the correlation of immune response as a readout. So this is the schedule of treatment. Just as mentioned, the patient will receive a total 4 shots within the first 6 months and carry on for 72 weeks for the follow-up. And we have dose escalation, and safety is a primary endpoint. So in summary, so SECOM, PSA and PSMA induced robust immune response in the preclinical models. Antibody from mice immunized by SECOM, PSA, PSMA recognize there's prospective antigen target, both in the cell line and in the tumor tissue. And the SECOM PSMA significantly slow the tumor growth in the mouse model and also prolong their survival. The PSA and the PSMA combination therapy can broke the self-tolerance in the non-human primates, and our ongoing phase 1 study is targeting for biochemical relapse high-risk patient population. Of course, all the study is in the collaboration with many individual and organization and teams effort. So I want to acknowledge the patient and the patient family, our study investigator and study site staff, and our preclinical and translational collaborators, and our clinical team at Inovia. Thank you so much.
Your preclinical data shows, which is good, that you increase interferon gamma as your marker, but we also know that interferon gamma actually increases PD-L1 expression. And I think we've seen a lot of vaccines or different vaccines approach that does it. So my question to you is, with the immune cycle and you may be increasing the antigen and the initial immune response, the breaks that may happen with immune checkpoints might basically blunt any effect you have. Are you planning any studies in the future to combine with immune checkpoint inhibitors to try to overcome that second break that'll happen after you induce the initial response?
Yeah, that's a very good question, and I consider this is your, very kind recommendation as well. So actually, we are in the planning of further study, look at the checkpoint inhibitors and associate the expression of PD-1, PD-L1, and other immunomodulators potential in the combination at different stage of the disease. Of course, in this particular study setting, in this particular patient population, maybe the combination therapy have their concern because this patient is relatively safe, healthier in the early stage, but I would definitely try to explore further for the different disease indication. Thank you. Sure.
I have one question about the target antigens and then one question about safety. So my friend Doug McNeil, who's sitting right there, has developed a PAP encoding DNA plasmid vaccine. He doesn't use electroporation, but it's otherwise similar. Why did you pick PSA and PSMA? And
well, that's different consideration. I mean, just as I said, we understand the association of a PSA, the PSMA in the disease of prostate cancer. Although there is some debating and controversial and back and forth, but we see the success in the PAP. And also we understand there's some attempt, at least, using the PSA, PSMA as immunotherapy target. So in this particular approach, we use the PSA, PSMA. So I know Doug have a beautiful achievement in the PAP, and I think he will mention later today. And so we try to see what is different between these, but the common goal is for our patient.
And the second question is about PSMA expression actually not being prostate cancer specific. And of course, it's found in the vasculature and in multiple tumors, and it's also found in the kidney. And one question is, have you guys seen any kidney toxicity in sort of your animal toxicology studies at all?
Well, we have the animal toxicity study in the animal models, and also we have a toxicity study in the non-human primates. And we have not observed any associate signal. And so we are very vigilant to look at both possibility in the clinical setting, including interleukin-12 related autoimmune possibility. So currently, the good news is that we haven't seen anything about it. Yes, Doc.
And follow up on that question, and also because I'm supposed to talk about targets, I guess, later. So in your preclinical study, the model that you're using in the tramp mouse and the C2, that's expressing mouse PSMA?
Yeah.
So
actually, I did mention, I give you the the chart of that things, we have the mouse specific construct. And we have, but we use the same construct as in human for non-human primates, but we have a separate mouse construct, which generated from PanLab. So, yeah.
Thanks.
Any question? Thank you.
Thank you very much.